The term glycolysis is derived from Greek words glykos meaning sugar and lysis means splitting, meaning splitting of sugar, mainly glucose. The order of events under glycolysis was given by three scientists, Gustav Emden, Otto Berov and J. Parnas, and therefore it is often referred to as EMP pathway also. In anaerobic organisms, glycolysis is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and it occurs in all organisms. In glycolysis, glucose undergoes only partial oxidation and the end product of glycolysis is 2 molecules of pyruvic acid per molecule of glucose. In plants, this glucose is obtained from sucrose and also from other stored carbohydrates, other forms. Sucrose is a disaccharide containing glucose and fructose. So it is first broken down into glucose and fructose, its component monosaccharides, by the enzyme invertase. And then these two monosaccharides, they enter the glycolytic pathway. Glucose is first phosphorylated to glucose 6-phosphate. Fructose is also phosphorylated to give rise to glucose 6-phosphate only. And the enzyme which does so is hexokinase. You can understand the name of the enzyme. It is activating by adding phosphate. What is it activating? It is activating a 6-carbon sugar, hexokinase. This phosphorylated form of glucose, G6P, isomerizes. This is the first step of glycolysis to form fructose 6-phosphate. Once fructose 6-phosphate is formed, the succeeding steps of metabolism are the same, whether glucose was the initial substrate or fructose was the initial substrate. Glycolysis is a chain of 10 reactions, each controlled by a different enzyme, and ultimately produces pyruvic or pyruvic acid from glucose. As we study glycolysis, note down the steps where either ATP is utilized or synthesized and step where NADH, NH+, are synthesized during glycolysis. This is the scheme of steps, 10 steps of glycolysis. Glucose, which is a 6-carbon sugar, uses 1 ATP. It takes its phosphate and energy to convert into glucose 6-phosphate. This glucose 6-phosphate isomerizes to fructose 6-phosphate. No energy is required for this. Fructose 6-phosphate uses another ATP and takes its phosphate and energy to convert into fructose 1,6-bisphosphate or biphosphate. This fructose 1,6-bisphosphate then gets broken down into 2 molecules of 3-carbon sugars. One is glyceraldehyde 3-phosphate or PGAL and the second is dihydroxyacetone phosphate or DAP. So 1 molecule of each, which keep isomerizing and turning into each other, as that step does not require any energy. The next step, the trio sugars, 3-carbon sugars, they lose molecules of hydrogen, hence NADH is produced and they turn into triose bisphosphate, that is 1,3-biphosphoglyceric acid each. So 2 molecules of 1,3-bisphosphate glyceric acid. The phosphate required at this stage is simply inorganic phosphate which is picked up from the cytoplasm and does not require any energy. So no ATP is used. And 1 NAD is reduced to NADH. So 2 NADH was produced because there are 2 molecules of triose phosphate per molecule of glucose. This 1,3-BPGA loses 1 phosphate and some energy which is trapped by ADP to convert into ATP. So 2 ATP are produced at this step, 1 ATP per molecule of 3-carbon sugar. 3-PGA isomerizes to 2-PGA, that is, the phosphate shift from the third carbon to the second carbon, requiring no energy. 2-PGA, two molecules of 2-PGA lose 1 molecule of water each to convert into 2-phosphoenol pyruvate. Phosphoenol pyruvate gives away its phosphate and energy to produce 1 ATP each. So 2 ATPs are produced, that is 1 ATP per molecule of triphosphate, and 2 molecules of pyruvic acid are finally produced. We can see that the process of glycolysis is complete, but the oxygen has not come into play till now. So process of glycolysis does not require oxygen as such. So we can notice that ATP is utilized. 1 ATP is utilized at 2 steps. First, for converting glucose into glucose-6-phosphate, and second, for converting fructose 6-phosphate into fructose 1,6-bisphosphate. Also, 2 molecules of NADH plus H are produced at the step where 3-phosphoglyceraldehyde is converted to BPGA. That is, PGAL is oxidized. Then 2 ATPs are produced, 1 ATP per molecule of BPGA, during conversion of BPGA to 3-phosphoglycic acid. This energy is trapped in the form of 2 ATPs. Again 2 ATPs are synthesized during conversion of phosphoenol pyruvate to pyruvic acid in the last step. Hence, we can see that during glycolysis, 2 ATPs are utilized and 4 ATP are produced for each molecule of glucose. In addition, 2 molecules of NADH are produced per molecule of glucose. So the net yield of glycolysis is 2 ATP and 2 NADH. You will learn later that if oxygen becomes available, each NADH can give 3 ATPs each. So let us summarize how many ATP molecules are synthesized directly from glycolysis from 1 molecule of glucose. As we have seen before, under anaerobic condition, glycolysis gives a net yield of 2 ATPs and 2 NADH. NADH has 2 main functions in cell. One, it may undergo oxidation in presence of oxygen to provide 3 ATPs. Another function is to provide H ions wherever required, for example in biosynthesis of some other molecule. Under aerobic condition, when oxygen becomes available, each NADH can undergo oxidation and yield 3 ATP. So under aerobic conditions, the net yield of glycolysis is different. It is 2 ATP and 2 NADH, which will give 3 ATP each. So net gain of 8 ATPs if oxygen is available. So glycolysis of 1 molecule of glucose has produced 2 molecules of pyruvic acid in the cytoplasm. What next? It depends upon the cell or conditions of the cell. Pyruvic acid, it can be handled in 3 major different ways. Lactic acid fermentation, or it can undergo alcoholic fermentation, or it can undergo aerobic aspiration when oxygen supply is there. Fermentation takes place naturally in anaerobic conditions in many prokaryotes and in some unicellular eukaryotes also. For complete oxidation of glucose, where it is broken down to carbon dioxide and water, the pyruvic acid has to undergo what is known as Krebs cycle, requiring oxygen supply, hence also known as aerobic respiration. Fermentation. The pyruvic acid undergoes fermentation, that is partial oxidation of glucose. There are 2 main types of fermentation, alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvic acid, which is a 3-carbon molecule, is converted into ethanol, which is a 2-carbon molecule, and 1 carbon dioxide molecule is given out. Hence the enzymes involved are pyruvic acid decarboxylase for removing the carbon dioxide and alcohol dehydrogenase to produce ethanol. This process occurs naturally in yeast. In lactic acid fermentation, Pyruvic acid is converted to lactic acid. This occurs naturally in some bacteria. It can also occur in higher organisms, for example in muscles during exercise when the oxygen supply to the muscle to cells cannot keep up pace with the metabolism occurring locally. The enzyme involved is lactate dehydrogenase, which reduces pyruvic acid to lactic acid. In both types of fermentation, the reducing agent, that is the H donor, is NADH, which as we discussed in the last slide, and NADH gets reoxidized to NAD+. And in both lactic acid and alcohol fermentation, not much energy is released. That is, less than 7% of the energy present in glucose is released. And even that is not efficiently utilized in production of ATPs. And both the processes are harmful. Either acid or alcohol accumulates which harms the cells. This is a diagram representing fermentation. Left side is the glycolysis resulting in molecules of pyruvic acid. Right side shows the pyruvic acid being converted to lactic acid in lactic acid fermentation, which is a 3-carbon compound, and the enzyme involved is lactate dehydrogenase. In the second type of fermentation, pyruvic acid is converted into ethanol and carbon dioxide. Pyruvic acid decarboxylase removes the carbon dioxide and alcohol dehydrogenase produces alcohol. We can see that the NADH, which was produced during glycolysis, is utilized for both type of fermentations. So the net result is only 2 ATPs. As we can see, in fermentation, the net ATPs synthesized are just 2. Yeast get poisoned when the concentration of alcohol reaches about 13%. Therefore, the maximum concentration of alcohol in naturally fermented beverages cannot be more than 13%, and additional processing is required if we require a higher percentage of alcohol. For complete oxidation of glucose, so as to extract a much larger number of ATP, aerobic respiration is required. This occurs in the presence of oxygen. The end products are carbon dioxide and water, and the large amount of energy which was stored in the substrate is released and it results into a larger number of ATPs. In eukaryotes, this aerobic respiration happens within mitochondria. So the pyruvic acid that was produced in the cytoplasm needs to go into mitochondria. In higher organisms, aerobic respiration is the most common form of respiration, and the next step in aerobic respiration, that is Krebs cycle, will be dealt with in the next part of the lecture.